Thank you all for coming today to our Friday seminar. It's really a pleasure for me to get to introduce Cagla today. We have known each other for a long time. We were both postdocs in labs that were kind of interested in how synapses form. And so there were really like 4 superstars in her lab that have all gone on to do great things. And we always kind of intermingled at meetings. And so there's just a lot of overlap between a lot of us in the Sains lab and Cagla, and I'll get to what lab she was in. So she did her initial studies, bachelor's and master's in Turkey, where she's from originally, and then she went to Germany to University of Heidelberg and the EMBL to do her Ph.D., and then from there took a long flight to Stanford, where she was a postdoc with Ben Barres for about 5 years. And in Ben's lab, she really pioneered some of the studies looking at how extracellular matrix molecules, particularly those that are derived from astrocytes, can influence synapse formation. And that was something that the Barres lab had really been doing for about 10 years at the time. And they had identified this molecule thrombospondin, and they didn't know how it worked. And so when Cagla was in the lab, it was her job to find the receptor for it, which was a gabapentin receptor. And so that was really what catapulted her to her own career. So that was a Cell paper. And while doing that, it turned out that other molecules from astrocytes could influence synapse formation. Some other labs found some, but Cagla also found a few others. A really interesting family that 2 different isoforms had differing effects on synapse formation, Spark and Spark-like 1 or Heaven. And from there, her career really took off. And so she has been at Duke now since 2008. She has way too many grants to list. But she also, in addition to being never enough, right? I think what's fascinating is that she has time for everything else. She helps to organize meetings, both internally at Duke, but also externally. She is a director of a graduate program there now in cell and molecular biology, and also a co-director of a new institute there on regeneration. So she's doing all these things, but her science still is really the best. I think one of the coolest things that came out of her lab, she actually sent someone to talk at one of our Glia meetings a few years. So she had been working on how astrocytes contribute to synapse formation and just neuronal structure in general. And then she found that some genes that we thought were only in neurons were actually made in astrocytes and were really important for astrocyte development and morphogenesis. And it's just been really cool to watch. So Cagla, it's a pleasure to have you here.
Thank you, Mike. That's such a kind introduction. And if you want, I can put on the mic, or I can. Is it better if I put on the mic because of the Zoom? That's really, really kind of you. It's a great pleasure to be here. I always think of Virginia Tech and this institute, also Blacksburg, as Glia Mecca, I call it, because there's so many people doing such fun stuff about the role of glial cells, particularly astrocytes, in health and disease. So it's a great pleasure to be here and share with you the work that has happened and is ongoing in my lab. So as Mike told you, I'm very interested in understanding how synapses are formed during development, how they are remodeled throughout life with experiences and learning, and as well as how they are maladaptively changed during disease states. And as a cell biologist, I wonder, oh, this won't work because I didn't. Does it work?
There's a button.
Okay. It might work now. Okay. As a cell biologist, our view of a synapse is composed of a cell-cell adhesion between neurons. You have a presynaptic neuron that makes an axonal bouton onto a postsynaptic dendrite, and the synaptic transmission occurs by simply the release of synaptic neurotransmitters that are then sensed by the postsynaptic receptors. But in recent years, we've expanded our understanding of the synapse to include a third cell type and a process that comes from it, the astrocyte perisynaptic astrocyte processes. These are both structurally and functionally intimately interacting with the synapse. They do express transporters and also receptors for neurotransmitters. So they can sense the neurotransmitter communication that is ongoing between the pre- and postsynaptic side. And in response to it, we know that they are undergoing both local and global calcium transients. We don't yet fully understand what those are encoding. But even more fascinating for me is that it's not that one astrocyte ensheets one synapse, it's numerically impossible, but an astrocyte ensheets and interacts with quite a large number of synapses. This is a mouse astrocyte. It interacts with around 100,000 synapses at the same time, therefore has the capacity to listen to and also react to all the ongoing local synaptic activity. We don't yet know how those interactions are contributing, that communication is contributing to the information processing power of the brain. But what we have been studying in the last 10 years and even longer in the field has been how do astrocytes contribute to the formation of these synaptic structures. And in the first part of my talk, I'm going to talk to you, first give you a historical kind of background on what we know about how astrocytes control synaptogenesis, and share with you an unpublished new work from my lab that is further understanding some of the interesting mechanisms of how astrocyte-secreted proteins control the formation of specific circuits in the central nervous system. And in the second part of my talk, I'm going to turn this interaction to the other side and show you some of the more recently published work that we found that neuronal architecture and neuron-astrocyte contacts themselves are extremely important for the development of this beautifully complex morphology of astrocytes. And in fact, if you impair that interaction between the neuronal processes and astrocytes, then you also impair the astrocytes' ability to control synaptic connectivity. So, quite ironically, much of the work that started the idea that astrocytes are important for synapse formation came from studies in neuron-only cultures. So to be able to understand the roles of the astrocytes in synapse formation, Barrett's lab developed a retinal ganglion cell culture system. These are retinal ganglion cells that are isolated from P7 red retina. They're completely pure. There are no other cell types in this preparation. And they can be cultured for at least 2 weeks and allowed to grow these beautiful neurite networks. And that way we can study synapse formation on these neurons. And we can also culture them then in the presence of astrocytes, and there are multiple ways we do that. One of the most often ways is that we culture the astrocytes on a feeder layer, which is on a membranous porous membrane. So it's not in contact with the RGCs, but the secreted factors can diffuse freely between the two compartments. On the other hand, we can also condition the media with astrocytes and then provide that media onto RGCs. And all of these are done in serum-free conditions in media where we know the exact composition of all the different factors that the neurons need to survive as well. So using these techniques, Barrett's lab around the end of the 1990s showed that, one other technical thing I would like to point out is that one of the ways that we can assay synapse formation is we stain for a presynaptic marker, which is in the axon, and a postsynaptic marker in the dendrite, and they would appear to colocalize at a synapse because they are at extremely close proximity to each other, because that's where the synapses are. But elsewhere, they would be in distinct compartments of the neurons, therefore they will not be colocalizing. So by quantifying the colocalization of those two markers, we can pinpoint where the structural synapses are. So, this is data from my lab, but also Barrett's lab, and many other labs have shown this, that if you culture neurons, in this case retinal ganglion cells alone, and stain for a pre- and a postsynaptic marker, you see some synapses, but not so much. On comparison, if you culture them in the presence of astrocytes, now there are many more synapses. And when you patch onto these neurons, you would also record much more synaptic activity. So, and in fact, all of the effects of astrocyte feeder layers can be recapitulated by simply transferring astrocyte conditioned media onto the RGCs, indicating that it's the astrocyte-secreted signals that induce formation and enhance function of synapses in this culture system. Now, I'm not showing you here today, but us and others have shown in many other preparations of neurons, as long as the neuronal cultures are pure, this is what you observe. You have a basal synapse formation between neurons in the absence of astrocytes, but if you provide astrocytes, you enhance this quite dramatically. So these astrocyte conditioned media then provided us a biochemical way to get at what are the secreted factors that are important for synapse formation. And it turned out that it is not just one type of molecule that induces synaptogenesis. Luckily for us, there are many different molecules that are doing really interesting synapse modulating functions. And in the simplistic terms, astrocytes induce formation of excitatory synapses at least two steps. The first step is they induce formation of what is traditionally known as a silent or a structural synapse. These synapses are pre- and postsynaptically, if you do EM, you see perfect pre- and postsynaptic sites, and if you do some fMDI experiments, you will see that their presynapses are secreting glutamate and recycling, but they lack AMPA receptors and they only have NMDA receptors on the postsynaptic side. So if you patch onto the postsynaptic neuron, you are not seeing an increase in the number of AMPA-driven postsynaptic currents. Astrocytes can then also insert the AMPA receptors onto these synapses and convert these silent structural synapses into fully functional synapses. And work from Barrett's lab first, and then later me in my lab and Nicola Allen in her lab, we've identified a number of factors using this system that induce these two steps. The ones that I'm going to talk about today are the thrombospondin family proteins. They were the first ones identified by Ben Barris's lab, and it's a family of 5 proteins. 4 of them are 1 through 4 are expressed in the nervous system. And as Mike also mentioned, my lab had worked on another family of proteins, particularly Spark-like 1. And we found that Spark-like 1 is also, in this culture system, induces the formation of silent synapses. Glypicans and chordin-like 1 are some of the other factors that come from astrocytes. They do insert the AMPA receptors to the postsynaptic side. And of course, in vivo, all of these work together to induce the formation of synaptic circuits. And I'll briefly tell you, so once we identified these factors and characterized them molecularly in culture, we also went in vivo, and particularly to a part of the cortex known as visual cortex, and focused our attention to the synaptic zone of the visual cortex, where there are many excitatory synapses, but also beautiful astrocytes that interact with them. And in this part of the brain, there are 2 major flavors of excitatory synapses. One are the synapses between cortical neurons, and we can capture them by the presynaptic VGLUT1 staining because that's found in the cortical neurons. And the second type of synapses are the thalamocortical excitatory synapses, which are positive for VGLUT2. And it turned out the thrombospondin family proteins are crucial for the formation and maturation of intracortical synapses, whereas heaven does not really do anything to intracortical connectivity, but primarily drives the formation of thalamocortical synapses. So So I'm not going to talk about heaven today and directly jump to thrombospondins. So how do thrombospondins control synapse formation? And this was actually the question that I answered during my postdoc, as Mike also mentioned, by looking for its receptor in neurons. And very surprisingly, it turned out to be not one of the canonical well-known receptors of thrombospondins, but a receptor that's also known as gabapentin receptor or calcium channel subunit alpha 2 delta 1. And I found that this protein interacted with thrombosponins, and that interaction was crucial for the induction of synapse formation. And a drug known as gabapentin, which binds to this receptor, and it's an anti-epileptic, anti-neuropathic pain drug, turned out to be able to occlude the binding of thrombospondin and therefore strongly limit thrombospondin-induced synapse formation. However, some of the things still remained unanswered because at that time we didn't have all the tools, such as conditional and full knockout mice. Because alpha-2 delta is present both pre- and postsynaptically, we didn't know if thrombospondin needed to bind to both of them or only one. And we also could not link any of the alpha-2 delta activity in synaptogenesis to simple effects on calcium channels. Because alpha-2 delta is traditionally known as an auxiliary subunit of calcium channels, we first hypothesized maybe it was the channel function or the amount changing that drove synaptogenesis, and that didn't turn out to be the case. More recently, Chris Risher, who is now has his own lab in West Virginia, he was in my lab as a postdoc, and he tackled this question and answered it really beautifully using alpha-2 delta knockout mice and conditional mice. And what he found was that, surprisingly, thrombospondin only needs the postsynaptic alpha-2 delta 1 to induce synapse formation. In the absence of alpha-2 delta 1, 50% of cortical synapses are gone, and the spines are extremely immature because the interaction between thrombospondin and alpha-2 delta 1, potentially mediated through an MDA channel function, recruits RAC1 activation to the spines and matures the spine structures by modulating actin cytoskeleton. So I hope I give you a background to also demonstrate that we now know a lot of things about thrombospondins and how they work. So maybe it's time to move on to a different molecule and not worry about thrombospondins anymore. You might say that. However, there are still very interesting questions about these proteins. And I'm going to tell you a related story today, which has been quite surprising for us and also extremely rewarding at the same time, to just show you how complex biology is, and not when there is a family of proteins, you should not assume they all do the same thing. So that's one of the first questions that has been remaining. There are 4 thrombospondins in the brain, and do they have different functions? Do they work at different synapses and have distinct synaptic properties? And there's still a question that we're trying to tackle, and I think we now have the best system to study that. We don't know if the release of thrombospondins are directed towards specific synapses, regulated through some neuronal interaction, and whether they need to be at a certain location to be able to work in their synaptogenic capacity. And these have been questions quite difficult to address. There are 2 reasons for that. One is thrombospondin expression in the cortex is extremely transient, and it really maps to that very small window, the second week in cortical development, when the spine structures are forming and maturing. And to address the location question, so just to remind you, this astrocyte, it's around 50 microns from one end to the other, and it is encapsulating 100,000 synapses. So how can we, within the structure, figure out where thrombospondin is being secreted to and what type of synapse it has next to it? And the cortical synapses are intermingled. There isn't really much of a good organization in this region to be able to address this question. The work I'm going to share with you is work by an extremely talented postdoc, Se-Won Ko, who came up with a cool way of addressing this. So there's actually a CNS structure which is very well suited for these questions because it is highly organized and its synapses are well organized, so you can correlate where thrombospondins are with potential roles for thrombospondins in distinct circuits. And that structure is the retina. and it's still part of the CNS. So retina has the photoreceptors, and then the interneurons and retinal ganglion cells. Remember, these are the cells where we and others identified and characterized thrombospondence to start with. And what's important is that right in between these neuronal layers, there are two beautiful synaptic layers where synapses are organized into even more precise sublamina. And we know this structure really well, thanks to work by Mike Fox and others. And we know what are the different neurons that pair up with other neurons. So the neuronal architecture is beautifully worked out. However, there is a major problem for us astrocyte biologists. There are no astrocytes within the neural layers of the retina. And that was the first reason we never considered retina to work on, because you stain for an astrocyte marker and you just don't see anything. But there's actually another cell, which is almost an astrocyte. I call it the supermodel of astrocytes. It's long and lean, and it has these beautiful processes that go into the synaptic layers and interact very intimately with synapses, very similar to what astrocytes do. These are the Muller glia. This is a radial glia that has pretty much after the closure of the neurogenesis periods, that pretty much takes over all the perisynaptic astrocyte properties in the retina. It is in green, you can see the processes that are infiltrating into the OPL and IPL and interacting very nicely with the synaptic layers. In a work what's the question was then, does Muller glia control retinal connectivity? And Sewon was studying retina because he was interested in understanding whether there were changes in synapse formation and function in retinal degenerative models. And while he was doing this, he stained retina, these are P30 ret retina, with antibodies against thrombospondins and their receptor alpha-2 delta 1. And what he found was, first, I was amazed at an age as late as P30, there's quite extensive thrombospondin expression in the retina. By using RNA in situ, he verified that they were expressed by Muller glial cells. And very interestingly, the expression of TSP1 and 2, they are not the same pattern. They are localized at different layers. TSP2 is enriched in the OPL, whereas TSP1 is more so in the IPL, where there are receptors everywhere. So Sewon said, then retina must be a good model system to study the distinct roles of thrombospondins and how they may be contributing to the formation of distinct circuits. And one other thing that he noticed was that thrombospondin 1 staining seemed to be having this interesting enrichment in 2 locations at the IPL. And this is important for someone who knows the IPL well, is because there are 5 synaptic layers in the IPL where we know which retinal ganglion cells project to and receive input from which interneurons. And TSP1 is localized to these synaptic layer 2 and 4. And that is the layer where a very important RGC subclass is projecting to. These are on-off direction selective RGCs. They are positive for CART, so we can label them in culture using a CART antibody as well. They receive inputs from bipolar cells, which are direction selective, as well as the amacrine, starburst amacrine cells for inhibition. So this got us excited, and then Sewon went back to his culture work, and because he did study TSP1 and 2 induced synapse formation in retinal ganglion cell cultures, so just briefly, we can make a single cell suspension from retina. We have methods to just purify retinal ganglion cells, and then we can culture them either alone or with purified thrombospondins, 1 or 2, and we've done this so many times, and every time we see that they have very robust synaptogenic properties. Anecdotally, we always thought TSP2 is more robust. So, for example, me in my postdoctoral studies, once I discovered this, I just stopped using TSP1 and just, focused on TSP2 because it seemed like it was a very robust synaptogenic effect. So I didn't put too much thought into it. But luckily, Sebon did. What he did is he went back to his data. He had many cells that he imaged, and then he plotted the population that responds to thrombospondin. And it turns out, when he does this, thrombospondin 1, RGCs, a population of them do not respond to thrombospondin 1, whereas another population very strongly induces synapse formation. On the other hand, thrombospondin 2 induces synapse formation in a more robust manner in all astrocytes, but in varying degrees. So they end up being similar when you take an average. So that made him to test then these two observations, whether the cells that responded to thrombospondin 1 could be these CART-positive on-off direction selective RGCs. And when he did his synapse assay, he included a marker for CART and quantified the number of synapses that are formed in response to TSP1 on these CART-positive cells. And I hope you can see they respond really robustly to thrombospondin 1, whereas CART-negative cells don't. And when he quantifies this, he found that TSP1 robustly induces synapse formation on CART-positive cells, but not thrombospondin 2 is promiscuous and induces synapse formation on both cells. So then that meant maybe there was something wrong with the direction selective circuit in the retina from thrombospondin knockout mice. And to test this, first he used staining. So one good thing about this layer is that the chat-positive amacrine cells project here, so you can use the chat staining to make a mask on this layer and then quantify the number of pre-postsynaptic puncta co-localizing. And when he does this and compares the wild type and TSP1 knockout mice, he sees a significant reduction in the number of excitatory synapses. So this is quite remarkable for me. I thought he would see nothing because remember there's TSP2 still in this retina. But despite the fact that there is TSP2, he is seeing a significant reduction. These findings also made us wonder whether the direction selectivity of the retina was affected. And to test this, we collaborated with Greg Field's lab at the Duke Neurobiology department. Lucky for us, he's really specialized on direction selective RGCs. And they have this really cool multi-electrode arrays where you can put a retina and then show the retina bright bars of moving in different directions and do a multi-electrode recording concurrently. And this way you can identify direction selective neurons. For example, this neuron that they are recording from is spiking when the bars are in this direction. And you can extract some electrophysiological properties out of these. One is the direction selectivity, so the direction this neuron is selective is this direction, but also the the width of this direction selective curve. So basically the sigma of the curve. The sharper the acuity of the direction selectivity, the smaller this arc should be. And the larger it is, it means the RGC is not that accurately predicting the direction. So when Suba from Greg's lab did this study, he found that both the wild type and TSP1 knockouts had direction selectivity, so there was no effect on that. However, the direction, the circular standard was increased, indicating that the acuity of the direction selectivity was diminished. So taken together, these data provided us a strong indication that thrombospondin 1 is required specifically for the formation of these direction selective synapses. But it also created a conundrum. So as I told you earlier, we know alpha-2 delta is there, and alpha-2 delta is the receptor for both thrombospondin 1 and thrombospondin 2. And it's also present in all that we can see, all the layers of the IPL. So how could thrombospondin 1 be specific to these cells and not thrombospondin 2? And as I mentioned, gabapentin is a drug that blocks the interaction between alpha-2 delta and thrombospondin. And it does indeed block it for not just thrombospondin 2, but also for thrombospondin 1. So if you give the RGCs gabapentin, regardless they are CART positive or not, they just don't form synapses in response to thrombospondin. So it is the obligate receptor for that. So that meant then there must be another receptor for thrombospondin that is working maybe as a co-receptor, but it confers the specificity. So to test if where the specificity comes from, say one went back to my old notebooks from like 2004, 2005, and I actually pulled up the notebooks and I was like, look at this, I kept notes. And then he was able to like go through my notebook, and I told him, you know, I chopped thrombospondin into smaller pieces, and I found that this little tiny fraction of the thrombospondin 1 and 2, they are both synaptogenic. And they are both also inhibited by gabapentin, because that's where the important interactions between alpha-2 delta and thrombospondin occur. So, but I thought there is all the rest of thrombospondin, potentially that's where it's conferring the specificity. He said, that's possible, but I would like to first test if these two are specific at all. And I said, don't even try, just go make a lot of other constructs and test the other domains. He didn't listen to me. Luckily, it turns out even this small structure already has the specificity in it, which made things much easier for us. But I hope you can see this synaptogenic domain of thrombospondin 1 is also specific to CART positive cells. It doesn't induce synaptogenesis in CART negative cells. SD2 is not specific. This also gave him a possibility of doing a little jigsaw puzzle between different pieces of thrombospondins to generate the chimeras. These two proteins are pretty similar. Domain structure is identical. Amino acid homology is quite high. So you can mix and match different domains, and you still produce really nicely the proteins. They are soluble, they are purifiable. So he made many different chimeras. I'm just showing you two important ones, because otherwise it gets really complicated. So in the first one, everything else is like thrombospondin 1, but just the first domain is switched with thrombospondin 2. And in the second one, everything else is like thrombospondin 2, but the first domain is switched with thrombospondin 1. And it turned out that first domain, the third type 1 domain, is the critical one. Because if you make this chimera, where the first domain is thrombospondin 2, rest is thrombospondin 1, it is no longer specific. In fact, it's like a super synaptogenic fragment now. And if you do the opposite, you take the promiscuous thrombospondin 2 and just put the thrombospondin 1 first domain, now it is specific. So whatever interaction is done by this domain is the one that's mainly driving the specificity. And that gave us a clue, because that is a domain that has been described to be an interaction site for integrin beta 1, an important cell surface protein and a well-established receptor for thrombospondins. So that was exciting. So at that point, we're very lucky to have people working on retina at Duke, such as Jeremy Kay. So Jeremy has been offering us help with this project already. So we turned to Jeremy and said, where are integrin beta 1 in which RGCs? And turns out it's really enriched in the direction selective on-off RGCs. And someone verified that by RNA-scope. I don't have that data. So it turned out integrin beta 1 is a good candidate to go after. So one of the ways that we can go after is use an antibody against integrin beta 1 to block integrin beta 1 interaction with thrombospondins and do our retinal ganglion cell assay. And when we did this, we found that thrombospondin 1 induced synaptogenesis onto these CART-positive cells was very effectively blocked. Interestingly, It's not blocking thrombosponin 2 induced synaptogenesis still. However, it does have a mild effect. Thrombosponin 2 is not as strong anymore. So it's possible that thrombosponin 2 can interact with multiple integrins, whereas thrombosponin 1, it requires integrin beta 1. So if you remove that interaction, it no longer can function. The opposite experiment was also done by Sewon. So he overexpressed integrin beta 1 in RGCs and then tested if this is sufficient to now make thrombosponin 1 work in CART-negative neurons, and that seems to be the case. If you overexpress integrin beta 1, then and these are CART-negative cells, they are blue because we have the blue fluorescent protein, but then SD1 is now, like SD2, able to induce synapse formation in these RGCs. So these culture studies indicated that integrin beta 1 is important for this circuit and is the receptor for thrombosponin 1. But Sewon went ahead and tested that in vivo using Cre-ER strategy. This cadherin-6 Cre-ER is expressed in these on-off direction selective RGCs, and combined with the integrin beta fluxed mice, we gave the tamoxifen relatively late, like around P9, P10, because integrins obviously are extremely important, not just for synapse formation, but for neuronal migration and outgrowth. So we waited for everything else in the retina to stabilize, and then just before the synaptogenesis period is kicking on and the thrombosponins are starting to work, Sewon administered the tamoxifen. And then we also have RFP reporter in these mice, so the Cre-expressing cells are TD tomato positive. So using that as a mask, he quantified the number of synapses that are formed in those layers, and you can see he saw a dramatic reduction in the number of synapses in the integrin beta conditional knockouts. So taken together, I hope I was able to show you today that thrombosponin 1 in the retina has this beautiful selective effect on the retinal synapse, on-off direction selective retinal circuit formation. And we are still investigating some of the questions that are outstanding. It's really interesting that we need to have this integrin beta 1 TSP1 interaction exactly at this location to confer the specificity. So how is that working molecularly? That's an interesting question to go after. If you remember from the beginning of my talk, the TSP1 was enriched in these layers. So how do Muller glia know to secrete it there? Or is it retained by integrin beta 1 there? That's something that is interesting for us to figure out if it is provided specifically to that circuit or it is enriched there due to its receptor. And those are 2 questions. Okay, great. So that brings me to the second part of my talk. So I talked to you about astrocyte-secreted factors that are controlling synapse formation. And now I'm going to tell you about how actually astrocyte interactions with the neurons and neuronal structures are regulating astrocyte development, and how impairing astrocyte development that is driven by neuronal contact can have detrimental effects of both astrocytes and synapses themselves. As I told you in the beginning of my talk, astrocytes are highly complex cells. In a mouse, an astrocyte can interact with around 100,000 synapses at the same time. And throughout evolution, astrocyte-like glial cells have always been complex. But I hope you can appreciate here that as the evolution, progressed and we came to human brain, the astrocytes became more complex and larger. This is kind of a in-scale view of a mouse and a human astrocyte. They're around 10-fold larger, and they do contact with around 2 million synapses, up to 2 million synapses at the same time. So it was very interesting observations for Jeff Stockstill when he started his Ph.D. in my lab. He really insisted on, studying this. How do astrocytes become morphologically complex? And Jeff has graduated in 2017, and now is a postdoc with Paolo Arlotta in Harvard. But Jeff, since we are developmental cell biologists, took a developmental approach first, used the ALDH1L1 EGFP mice, and again focused on the visual cortex, and determined what is the timeline of astrocyte development using these mice. And I'm not going to show that data, but in principle what he found was that the astrocyte numbers more or less stabilized in the period between P1 and P4. That's when they divide and they align themselves along the cortex. But after P7, the number of astrocytes didn't change, but there was this green haze that was increasing in these cortices because the astrocytes started to infiltrate into the neuropil. And at a cell-specific level, he could image and visualize this by using a technique known as postnatal astrocyte labeling by electroporation. It's like in utero electroporation, only simpler. We take the P0, P1 pups, inject the plasmids into their ventricle, apply current, and then sparse populations of radial glial cells take in the plasmids. At that point, they are making astrocytes, so it goes into a low number of astrocytes, and then we can visualize those astrocytes and determine their structure. And when he did this for P7 versus P21 astrocytes, what he found was that the way they infiltrate the neuropil is greatly enhanced. So you can take a little cube in its domain and quantify how much infiltration it has into that cubic space. And as you can see, between P7 and 21, there is a great enhancement of that. And it can be measured by a cytosolic soluble EGFP or a membrane protein M-Cherry-Cax. And he also determined the territory of the astrocytes greatly enhanced by doing the quantification of the domain's size of the astrocytes. And since this is visual cortex, we can apply some fun tricks with experience. What he did, he raised some mice in complete darkness, so they never actually experienced any vision. And when he did this, he saw a remarkable reduction in the neuropil infiltration and also territory size of the astrocytes, indicating that neuronal activity changes and potentially neuronal developmental changes affect the astrocyte morphogenesis. So to understand the molecular mechanisms that may underlie this process, he wanted to develop a simple culture system so he can, test the genetic genes that are important in this. And for that, he isolated neurons from rat brain as well as astrocytes separately. And then he cultured the neurons by themselves first. And then he transfected the astrocytes with a fluorescent marker and put it onto the neurons. And when you do that, you'll realize that the astrocytes that are cultured by themselves or cultured on COS7 cells, and these are astrocytes that are purified by the very standard, very old-fashioned manner, because they've been cultured from P0 brains and they've been in serum before. Even though you take off the serum, they remain quite immature. But if you put them onto neurons, they, within 12 to 48 hours, take this much more complex structure, and this gives us a really good readout so we can see the effects of different manipulations on astrocyte morphogenesis. So he characterized this culture system really well and determined that it wasn't neuronally secreted proteins that drove this. It was not too much affected by blockers of synaptic activity. And he did the following experiment, which I think is quite fun. He fixed the neurons, so now the neurons are dead, but they are structurally there. And then he put the astrocytes on top, and the astrocytes still became morphologically complex. So it didn't even require live neurons for this to work. They do look different, so it's not identical, but pretty much complex. So what these told us that it was predominantly the adhesion-based mechanisms in this culture system that drove the morphogenesis of astrocytes. And what are they adhering to? We think it's the synapses, because the astrocytes love synapses. And you can see here in this cultured astrocyte with the neuronal synapses marked with Bassoon and Homer, this astrocyte is so much more complex where the synapses are and far less complex where there are no synapses. So then he hypothesized maybe it's the synaptic cell adhesion molecules that regulate astrocyte-neuron synapse association by driving astrocyte complexity. And we know a lot about neuronal synaptic adhesion proteins, many, many years of work. Also, in our own lab, we've investigated the roles of adhesion molecules at the synapse that bridge the pre and post. And in particular, Jeff wondered whether any of these molecules could be important for bridging astrocytes and neurons. And in particular, he was interested in the neurexin neuroligin family because he worked together with Sandeep Singh and also, again, a fabulous previous postdoc in my lab. They found that spark-like-1 or heaven binds to neurexin and neuroligin. That's how it forms thalamocortical synapses. But while Jeff was working on that with Sandeep, he also noticed that neuroligins are not only expressed by neurons. In fact, those were the times when all these very nice gene expression databases were coming out, both from Barrett's lab as well as from Balkak's lab and now from Nicola Allen's lab. These have repeatedly shown that neuroligins, in this case neuroligin 2, is highly expressed by astrocytes at levels higher than or at least comparable to what it is in neurons. But so until that point, majority of the work, if not all, was done on the roles of these proteins in neurons. So he wanted to ask whether neuroligins would be important in astrocytes to mediate astrocyte morphogenesis. And to test that, he used shRNAs against neuroligins, and there are neuroligins 1, 2, and 3 in astrocytes. Jeff, of course, went ahead and verified this both by protein and mRNA in the mouse brain and in rat astrocytes. And what he found was that if he uses an shRNA against neuroligins, he can completely abolish the morphogenesis that is induced by astrocyte-neuron contact. So it's quite a dramatic effect. In fact, each protein, if you silence each one independently, it has an effect. It's not as severe as the full knockdown, but in each case, you can put back a cDNA that expresses an shRNA-resistant form and rescue this effect. So this showed us that each neuroligin is important, at least partially, for astrocyte morphogenesis in vitro. We could also use this rescue strategy to test whether the extracellular region of neuroligins was important. So neuroligins have these large cholinesterase-like domain extracellularly, which interacts with neurexins. And there is a construct that was made by Peter Scheifele's lab where they swapped the extracellular region with cholinesterase itself. And this no longer binds neurexins, but it makes a nice protein that goes to the cell surface. So it's called neuroligin-1 swap. This swap protein, even though it was expressed on the membranes of the astrocytes, it could not rescue the morphogenesis, indicating that it is the interactions by the extracellular sites, potentially with neurexins, that is what is inducing this morphogenesis. So Jeff went ahead and knocked down the expression of neurexins in neurons and found that to cause astrocyte morphogenesis deficits, even though the astrocytes still contain neuroligins, giving us a clue that the neurexins were required. However, that experiment did not address the sufficiency. So he addressed the sufficiency with the following experiment. So he generated a meshwork to mimic the axons using nanofibers and then coated these nanofibers with different neurexin extracellular domains. So just making the fake axons. In this case, there are no neurons anymore. We just have fake neuronal networks here. And then put the astrocytes on top. And what he found, this is the control situation where he just has FC tag. And then the astrocytes is interacting with the nanofibers, but not so much. And then when you have different neurexins there, you now start to induce the arborization of astrocytes. And of course, there are also alpha neurexins. There's potentially extremely enriched complexity here. So it's possible that neuroligins and astrocytes could direct astrocytes towards specific synapses depending on which neurexins those synapses are interacting. So these in vitro experiments gave us a clue about the molecular mechanisms of how this is working, but we also wanted to understand in vivo what is the function of neuroligins in astrocytes. And to test that, he again used the postnatal electroporation strategy and put in the assateranase against the mouse neuroligins to see into the astrocytes at P0 and then determined if this affected their structure at P7. He measured the neuropil infiltration volume, and he found that neuroligin 1 and 2 silencing significantly impairs infiltration into the neuropil. At this stage, neuroligin 3 knockdown doesn't, but when he waited 2 more weeks and did the same analysis at P21, surprisingly, SH neuroligin 1 expressing astrocytes caught up with the wild type, whereas neuroligin 2 or 3 silencing greatly stunted astrocyte morphogenesis. And here is a quantification of that with respect to the developmental time point, and it shows you that either neuroligin 1 or 3 silencing robustly stunts growth of astrocytes between P7 and 21. Because neuroligin 2 was so important both early and later, we decided to focus on neuroligin 2 for the rest of the study. We acquired a fluxed allele of neuroligin 2 and also included a tomato Cree reporters, so we can label the cells that are knockout, and then used PALE to introduce Cree plasmids into sparse populations of astrocytes, and then look for the astrocyte morphology at P21. And just to show you with staining that this in this mice, we can decrease the neuroligin 2 specifically in astrocytes. Neuroligin 2 staining is still present in non-astrocytic structures, which is what you expect because this is a conditional manipulation. And then when we analyze the astrocyte morphology, this is a wild-type astrocyte, very robustly filling the neuropil. This is a het astrocyte. Now, we already see an effect of just losing one copy, and if you lose both copies, now you have a more dramatic reduction in astrocyte morphogenesis. So taken together, this showed us that neuroligins in astrocytes, neuroligin 2 particularly, is important for astrocyte morphogenesis. But how does this affect synapse? And one possibility is that this could also change the synapse numbers within the domain of that astrocyte. As many of you may know, the astrocytes have their domains, and next to it there is another astrocyte, and each astrocyte is responsible for the synapses within their synaptic island. And we could use this to our advantage to figure out how the synapses within the domain of this manipulated astrocyte compares to its next door neighbor, which is essentially wild type. And when you stain the astrocytes that are manipulated, this is blue here, and pre- and post-synaptic markers, then you can go in and quantify the number of synapses that are within the territory of this astrocyte with respect to the next astrocyte, which is wild type. So when Jeff did he used vGlut1 and Ph.D. 95 for intracortical synapses, and very surprisingly, he saw a really dramatic reduction in the number of excitatory synapses within the domains of knockout astrocytes. This replicated also for the vGlut2 Ph.D. 95 positive synapses, a dramatic reduction there as well. So telling us that the loss of expression of neuroligin 2 in astrocytes affected predominantly the excitatory synapse numbers in their domain locally. We were expecting to see some changes with inhibitory synapses, but in fact we couldn't find any difference between the domains of the knockout or their neighboring astrocytes. So this was surprising because in neurons, neuroligin 2 is mostly considered to be at the inhibitory synapses and affect predominantly inhibitory synapse numbers and activity. But in the astrocytes, clearly it has a distinct function. So to get at this at a physiological way, of course, we can't just use these very sparse knockout approach. But at that time, we had this GLAST-3 ERT2 mice, which is able to very specifically manipulate neuroligin 2 only in astrocytes. So we've done all the specificity tests, and it's 100%, almost 100%. I think we found only 0.2% of the times it hit a neuron, so it's extremely astrocyte specific. However, that extreme astrocyte specificity in the cortex comes with the fact that it's a mosaic, so some astrocytes are not hit because it's a Cre-ERT2 system. So we characterize this, it's around 55% of the astrocytes lack their neuroligin 2. And we collaborated with Ru Rongji's lab at Duke, and when they patched onto layer 5 neurons, which have these beautiful dendritic trees that touch every layer at the cortex, so they have a lot of chances to interact with these knockout astrocytes. When they did the recordings for mini EPSCs and mini iPSCs, they found that mini EPSCs were indeed affected in these mice. They were reduced, and the amplitudes were reduced as well. And surprisingly, they also found that mini iPSCs were enhanced. So even though we didn't see a change in the number, the activity potentially of the inhibitory synapses were also affected. But taken together, this showed us that by only manipulating the astrocytic compartment of the tripartite synapse, it is sufficient to change the inhibition and excitation balance in the cortex. So that brings me to my conclusion slide. So we found that astrocytic neuroligins interact with neuronal neurexins, and this interaction is important for controlling astrocyte morphology and synapse formation. Of course, we're following these up. We want to understand how astrocytic neuroligins control the astrocyte morphology. Are they affecting the astrocyte actin polymerization? But also, how are they controlling synapse numbers? That's quite curious for us. How does this contact-mediated mechanism is regulating synapse numbers? And this also gave us a lot of new avenues to go after. I mean, neuroligins are just one class of cell adhesion molecules on astrocytes. We went in and used the same in vitro and in vivo tools to screen for other cell adhesion molecules in astrocytes and identified a number of them as important regulators of astrocyte and also synapse development. And importantly, are these pathways impaired in disease? What I have forgotten to tell you, both neurexins and neuroligins are linked to neurological disorders, including schizophrenia and autism. And most of the time, as I said, they're studied for their roles in neurons. But the synaptic abnormalities could be originating also from astrocytes. So understanding their roles in astrocytes is fundamentally important for us to decipher their roles in synaptic pathologies and diseases. This brings me to my favorite slide. I have to thank all these fantastic people that are working with me and have worked with me. Jeff is now doing his postdoc. I highlighted the work by Se-Won Ko today, as well as Jeff Stockstill. Jeff was working closely with a current graduate student in the lab, Juan Ramirez, and another postdoc, Katie Baldwin, and the ones that are highlighted in yellow here are the mini lab that Se-Won runs within my lab, all his trainees, and they're all doing great work. And thank you very much, and I look forward to your questions.
Time for a few questions.
Since you introduced the last part of the talk with this evolutionary difference that induces this higher level of complexity in the astrocytes, did you consider looking at the pairing of human neurons with rat astrocytes, and or looking at whether the neuroligins are actually changing during the course of evolution?
Yes, that's a fantastic question. Yes, we would love to do those experiments, and we would like to start doing the chimeras, like put the glia into the mouse brain. And I don't know if you're familiar with Mike Niedergard and Steve Goldman's work, where when they implant these glial progenitors into the mouse, from humans into the mouse, they take over. And why are they taking over? Are they more aggressively going after synapses, right? So there could be two things. One is they have more neuroligins or neuroligin-like molecules. And also, in addition, maybe they have more cell adhesion molecules on them that normally mouse doesn't interact, but these cells can. So that's definitely something that we would love to work on. The other cool thing is, right around the time we were publishing our paper, actually theirs came a few months earlier than us, so they've isolated fibroblasts from schizophrenia patients and controls, made glial progenitors, injected these progenitors into mice, and then found that the glial progenitors from schizophrenia patients induce some behavioral abnormalities. But before transplanting them, they did an RNA-seq to see what were the major differences between glial progenitors that came from schizophrenia patients versus controls. And they found a great reduction in the levels of cell adhesion molecules, including neuroligins. And so that was kind of nice. And they have a line in their paper. One wonders what these otherwise synaptic protein, thought to be synaptic proteins are doing in glia. I think this is probably a common theme for not just neuroligins, but any molecule that we may have been studying in neurons, thinking that it's only in neurons, but the astrocytes use them too, to either sometimes compete or also to collaborate with the neurites to form these circuits. So I think we need to expand our understanding of these synaptic adhesions, or these proteins being just synaptic adhesion proteins. I think they're just adhesion proteins between cells, and then the cells utilize it and for competition or cooperation so that they can put together a very robust nervous system.
So in neurons, the neurexins complex with several other proteins like municipalities, as well as Cas.
Mm-hmm.
So you showed the data where you have just the extracellular domain of neurexin inducing this astrocyte complexity. I was wondering if you looked in vivo or in culture at maybe the complexes formed by neurexin when it binds an astrocytic neuroligin versus when it binds a neuronal neuroligin, and how that might be different or similar.
That's a great question. So that's definitely part of our plan, because we're trying to figure out how the neuroligin in astrocytes is affecting synapse numbers. And, it should be, at least in part, mediated through neurexin interactions. One of the experiments we've done, we've generated synaptosomes from neuroligin to conditional knockout mice to look for what we're missing at the synapse. And neurexin 1 is missing. It's not completely gone, but greatly diminished in amount. So one possibility that we're thinking is that by generating a neuroligin cloud of astrocytes around the synapse, it might be capturing the neurexins between the neurons rather than diffusing out. That's one possibility, and that seems to be important for that. The other thing that's very interesting is we see reduction in NMDA receptors, and again, that could be maybe now thrombospondins or heaven is not directly coming to the synapse, and it's because now the astrocytes retracted, so now the NMDA receptors are diffusing out or off the synapse. So those are definitely some of the things to think about.
Getting back to Harry's point, sort of from an evolutionary perspective. So, first of all, do T-Leos fish have an equivalent homolog of thrombospondins in them? And then I can't help but think about this lifelong developmental gradient in the T-Leos retina, where as you move out into the periphery and it's growing through life, you get these different types of directionally selected retinal ganglion cells that are basically telling the fish about things swimming by them in the periphery. So my first question is, do they have the thrombospondins?
They do have thrombospondins, and they do have 1 and 2. They must have a homolog of 3 and 4 and 5 as well. So 3, 4, and 5 are a bit different than 1 and 2, because they lack the domain that I showed you. 1 and 2 evolved later, but the fish has it. And I don't know if fish has multiple forms, because fish always have like multiple of everything. So yeah, they might even, actually it's a good idea to check the fish, because they have a much better on-off, their visual system. Yeah, yes.
And they have, the gradient of Muller cells in the fish nice system to look at statically at a given stage of development. So the animal's older, it's not in early development, but the retina has a developmental gradient across it from central to periphery. And the Muller cells, I remember some stuff on Muller cells with development eccentricity in fish. There's some really interesting things in nonlinear interactions. I had no idea about the respondent effects as you go peripherally.
Yeah, no, that's a great point. Thank you.
One more question.
So you mentioned that thrombosponin has high level of expression during relatively short time window in development. So my question is how astrocytes maintain synapses in development versus in adult brain? In other words, are there different molecules or the way how they interact when you compare developmental signaling versus plasticity in adult?
That's a great question too, and it has been super challenging to study using antibodies because the antibody staining I showed you is from rat retina. We can see the protein in rat retina. If we try to stain the mouse retina, we just cannot stain it. Antibodies are having a huge difficulty, and so we recently developed a method to CRISPR knock-in tags into proteins that are in astrocytes. We're trying to see if we can do it in the Muller glia as well. So when we did it in astrocytes, we can still see some thrombosponin signal later on, but it's much, much more diminished. So it is possible it's there. It's possible it's secreted and degraded very rapidly. Actually, yesterday a paper came out from Balkak's lab where they found TSP1 is upregulated when they give a CNO-induced activation of astrocytes in the striatum. So it's possible that they are there, but they are only provided for need basis, so to potentiate some synapse or so. That said, thrombosponin 3 and 4 mRNAs are present in the adult brain. Thrombosponin 4 is really highly enriched in white matter astrocytes, but the mRNA is also present in the gray matter astrocytes. So, and it is localized to the perisynaptic processes. So it's possible that TSP4 is the one that is kind of a maintenance factor. TSP3 also is there in adult, and it's very understudied. It would be interesting to see that one. So yeah, I think we need better tools, and I'm very excited about the CRISPR because it gives us the way to tag the original genetic loci and not depend on really bad antibodies. Yeah.
Synaplogenisis in adult brain,
Yeah.
for example, after stress or cocaine or so forth, there are formation of silent synapses. So I was wondering if mechanisms in adult brain are different or same.
Yeah. Yeah, so definitely. We are using the alpha-2 delta knockout mice, the conditional knockout mice, and finding that that receptor itself is regulating very interesting behavioral paradigms. So now the question is it binding thrombosponins, and which thrombosponin or thrombosponins? So definitely. But one thing I can say, as soon as there is an injury or maybe a systemic LPS challenge, the astrocytes, as soon as they become GFAP positive or reactive, they upregulate TSP1 and 2. And that's a very dramatic upregulation. And you can see it by Western blot. If you had a good antibody, you would see it by staining. But I think at a steady state, there might be something with that they are provided and quickly cleared out. But that's not the case in the retina, which is also interesting. That means you continuously need that signal to maintain that circuitry. Yeah, thank you.